We thank you all for joining on a beautiful and warm Friday, at least here in Ann Arbor, Michigan. Feels like summer after our Memorial Day weekend last week. We will be going, as we did last year, on a bit of a summer holiday. So we'll resume these in September. We do have talks scheduled for the entire fall, I believe. So, but we're always still looking for good presentations or talks. So please reach out to Renee or to me if you'd like to nominate yourself. Otherwise, we'll come find you based on talks we see. We've been looking forward to this talk today for a while now. I've known about Kelly's research, and I got to see a little bit of it at IAGLR, I guess 2 or 3 weeks ago now. And she's going to be providing even more information if you attended IAGLR and got to see her talk during the symposium that Aaron Dunlop and Andrew Muir pulled together. And so her talk today is about 50 minutes, but she, just to give everybody a heads up, so it's going to be packed full of great information. We do hope that you'll stay on for questions and discussion afterwards, if you do have the time to allow that. A little bit of background about Kelly. She's originally from Pennsylvania, went and got her undergraduate degree at Boston University in biology, with the specialization in conservation biology. She got her master's degree at University of Central Missouri, looking at wastewater effluents on Ozark streams, particularly with mottled sculpin, sort of that target species. And since 2020, she has been working on her PhD on the topic you will see today. And her advisors are listed there, Scott McNaught and Tracy Kalerowitz at CMU. And she tells me she hopes to become Dr. Kelly Hoyer by next May. So with that, thanks again for joining us, and we look forward to your talk, Kelly.
All right. Well, thank you, Bo, for that introduction. And thank you everyone for tuning in to my talk. I'm very excited to speak to you all today about pretty much almost everything I've been working on for the last 3 years now with my PhD research at Central Michigan University. And I'd like to start off by acknowledging my co-authors, Jory Jonas from Michigan DNR, Jason Smith from the Sault Ste. Marie Tribe, Chippewa Indians, Scott McNaught and Tracy Kalerowitz, also from CMU, and Ellen Marson, the University of Vermont. And so I'm going to be talking about the work I've been doing on larval Lake Whitefish and Cisco. So that I'd also like to acknowledge the fact that this is a highly collaborative project. Many agencies and universities have collaborated on this and are involved in multiple aspects of this overall project. So without further ado, I will get right into it. I'll keep the background brief because I know most of us are familiar with these two species right here. Lake whitefish, Coryglonis clupiiformis, and Cisco ciartidae are two closely related salmonid fish species native to the Laurentian Great Lakes. And not only do they serve as really important energy transfers throughout the food webs, but they also serve an economic importance of the fisheries throughout the Great Lakes. They also are very culturally valuable to the many Native American tribes that live around the Great Lakes region. Lake whitefish and Cisco exhibit pretty similar early life histories. So starting in mid-March or early April and until early summer, so around mid-June, when they'll go out to deeper waters, they'll be found in these nearshore sandy substrate habitats where they will start feeding on small zooplankton items like copepod nauplii, small cadacerans like this chiodorid right here. Then they'll start consuming larger cladocerans like Daphnia and adult copepods. And then they'll move on to macroinvertebrates like this midge larvae right here, and are exposed to similar nearshore introduced predator species such as the rainbow smelt and the round goby. So kind of setting the stage here for what I'm going to be talking about throughout this talk here. So throughout the decades, both species have experienced pretty wild changes in population sizes due to stressors such as overexploitation and predation by invasive species such as the sea lamprey, and environmental stressors all kind of working together against them. The decade that I want to focus on for right now is the 2010s, specifically 2011, as this is when Cisco began to exhibit a resurgence in northern Lake Michigan. As we can see from this Claremont et al. 2019 study here on the left, we're seeing increases in larval Cisco densities throughout the early 2000s, and then that's reflected in the commercial harvest here, here on the right. So throughout the 80s and 90s, we see pretty steady low numbers in adult Cisco, and then once we hit that to the 2011 year, those numbers start to skyrocket. However, that has not been the case for Lake Whitefish. In fact, they have continued to decline in recruitment in the same region. And this has led to a bottleneck in Lake Whitefish beginning during that first year of life. Still unsure of whether that starts at the larval or the juvenile stage. So the overarching goal of my dissertation research is to figure out what factors are at play in why this discrepancy is occurring between recruitment levels in these two species. And there's a lot of factors that could be at play here, such as reduced prey, as well as altered prey composition, predation by introduced nearshore predators like the rainbow smelt and goby that I mentioned briefly, reduced growth and recruits per kilogram spawner biomass, other unknown factors, climate change, other environmental stressors that could be at play here. So my job here is to take a few potential factors and figure out whether or not this is possibly contributing to these different recruitment levels. So I just want to briefly give an outline of what I'm going to be talking about for the rest of this talk. So first, I'm going to be going over metabolic rates, kind of current info for what we have right now on larval Lake Whitefish and Cisco, and what I've done to compare these metabolic rates at the larval stage between these two species. And then I'm going to be talking about foraging success, so talking about the lab studies that I worked on with Lake Whitefish and Cisco with four different prey items and seeing how their foraging abilities compared. And then I'll be talking about some field observations from our collected field samples and talk about differences in growth between Lake Whitefish and Cisco that we're seeing, as well as different prey compositions that we're seeing out in our sites that we have. And then last, I'll be talking about predation avoidance and how they could be different between Lake Whitefish and Cisco, how different differences in vulnerabilities to the rainbow smelt and the gobies make their merry way here. So first off, I'm going to be talking about metabolism. So how does routine metabolism affect larval fish growth? So we know generally that metabolism will increase with temperature. You can see from this figure down here, temperature goes up, metabolic rate goes up, and that can either increase or decrease growth depending on how much food is available at a particular area, since an increase in metabolic rate does require more food intake to sustain growth. So there is an optimum temperature at which and a feeding rate that will be optimal for growth in fish, but that prey availability has to be there. Also, weight-specific resting metabolism decreases as body size increases. So current information on carrageenan metabolic rates is pretty limited, especially at the larval stage. There have been studies done on Cisco and Lake Whitefish at the juvenile stage and older. And I've primarily found active metabolism, not a whole lot on routine metabolism. So this Bernatchez and Dotson 1985 study looked at juvenile Cisco and Lake Whitefish and found Cisco to have lower metabolic rates, but they did kind of give a disclaimer there that the chamber sizes as well as velocities, so since they were doing active metabolism, different velocities in the chamber to sustain swimming speeds were not standardized. So they did mention that that could have attributed to the differences between the two species as far as active metabolism went. And then Dabrowski, 1986, did trials on juvenile carrageenans as well. So not a lot of information available on larval, Lake Whitefish and Cisco in resting metabolism. That's where I wanted to fill this knowledge gap by completing some trials of my own here with metabolic rate and comparing them between Lake Whitefish and Cisco at 2 different sizes and at 3 different temperatures. So for my respiration experiments, I used a fire-sting oxygen probe equipment. So this apparatus here has 4 oxygen sensor channels that connects to a sensor, and then the sensor goes into a oxygen probe holder that you'll be able to see in my next slide. And it also is equipped with a temperature sensor, so that will log temperature throughout the entirety of the trial. And this is connected to a laptop, which is equipped with PyroScience oxygen logger software that will read oxygen levels throughout every second throughout the duration of the trial. So these are on the left here is kind of what you can see the entire chamber with the probe holder, and the probe is in the chamber, and it's completely airtight. And on the right is kind of a close-up of the chamber, and you can actually see the fish in there. So chambers were placed in water baths for each trial to control for temperature. For each trial, I had a control without any fish in there to account for oxygen being lost, not due to any fish consuming it. And I conducted these trials at 3 temperatures, 8, 10, and 12 degrees Celsius, and then at 2 sizes, small and large. And after each trial, I got standard length and dried out the fish for 24 hours to get dry weights. For data analysis, I regressed oxygen depletion against time to get a slope here, and then calculated metabolism in micrograms of oxygen per microgram of fish per hour. And after correcting for controls to make sure that I was getting the accurate numbers here. And then I conducted an ANOVA, or analysis of variance, to compare routine metabolic rate between Lake Whitefish and Cisco across 3 temperatures. So these are my results here for routine metabolism. So Cisco is in the orange, Lake Whitefish is in the blue, and we have 3 panels here, 8, 10, and 12 degrees Celsius for temperatures. And then at the left-hand side of each temperature panel is the large size, and then on the right of each panel is the small size. So we can see that small Cisco had significantly higher metabolic rates than Lake Whitefish at each temperature. And whereas large larval fish didn't exhibit differences in metabolic rate at each temperature. But with increasing temperature, metabolic rate increased for both species at both sizes. So kind of to summarize this so far here, small Cisco, we're seeing higher metabolic rate at all temperatures, which could lead to faster or slower growth depending on food availability that we're finding out in the field. So that could indicate a spatial component, and we'll come back to this later as we talk about our field results. And as temperature increases, metabolism increases in both species, which is consistent with what we know about how metabolism works with temperature. So to kind of take us into my next topic here, Cisco having higher metabolism, depending on where we're finding them in the field, could give them an advantage in terms of getting more prey than Lake Whitefish. There we go. So the next thing I'm going to be talking about is my foraging trials. So the goal here was to compare foraging abilities between Lake Whitefish and Cisco at 4 different prey types at 3 different sizes. So I hypothesized here that Cisco would have higher capture rates and lower handling times of prey items at lower densities than Lake Whitefish, as this could be an explanation for these recruitment differences. So for my short-term foraging experiments, I had 6 larval fish per 3-liter tank here. And I gave them one of 6 densities of 4 different prey types. So the prey types I chose were cyclopoid copepods, our representative copepod, Artemia nauplii. And I chose Artemia nauplii because they're analogous to copepod nauplii, and which is an important prey item for larval carrageenans. And they're just a lot easier to obtain in a lab setting than it would be to get them out in the field. So that's why I chose those. And then Bosmina was our representative cladoceran taxon, and then chironomid larvae was our macroinvertebrate. And I conducted these at 3 different sizes, small, medium, and large, for a half an hour for our small fish, and then for 15 minutes for the other 2 sizes. I use functional response analysis in R or Frere model to get a functional response type. So it uses logistic regression to determine that. So my goal was to get a type 2 functional response where prey consumption increases proportionately with prey density until an asymptote is reached, and at which point the consumption no longer increases with increasing prey density. And Frere uses Ryder's equation, which accounts for prey depletion, because I didn't replenish prey throughout the trials. And Frere calculates 2 parameters, so handling time and then resource capture rate, and conducts a difference test to determine whether there are significant differences between Lake Whitefish and Cisco in either of these parameters. So these are my results for small Lake Whitefish and Cisco. So the only prey item that there was a difference between the 2 species is Artemia nauplii. So Cisco had a higher capture rate of Artemia as well as a lower handling time. And all other prey items, there did not exhibit any differences between the 2 species. Except I guess you could say Bosmina. You could notice the blank space here where Cisco didn't actually eat enough Bosmina to generate a functional response curve. So I guess you could say by default, small Lake Whitefish won out there on Bosmina, but cyclopoids and chironomid larvae didn't show any differences. For medium carrageenans, medium Lake Whitefish had higher capture rates of all prey items than Cisco, and lower handling times for mostly all prey items than Cisco. Aside from chironomid larvae, the handling times here were pretty much even between the 2 species. But medium Lake Whitefish beat out the Cisco here for all prey items. And then for my large carrageenans, we had Cisco have a higher capture rate for cyclopoid copepods, as well as a significantly lower handling time. And whereas Lake Whitefish are still having higher capture rates of Bosmina than Cisco. And for handling time, Cisco had lower handling times at all prey items. So you can see how many chironomid larvae they both ate. So you really see they really start to go to town on those chironomid larvae as they grow, which is pretty cool to see. So to kind of summarize this up, small Cisco outperformed Lake Whitefish on Artemia nauplii, whereas medium Lake Whitefish outperformed Cisco on all prey items. And then large Cisco had outperformed Lake Whitefish on cyclopoid copepods, but Bosmina had higher success than Cisco with the Bosmina prey item at small, medium, and large sizes. So now I kind of want to talk about prey densities that we're finding in the field and how they've become pretty low over the years, and how that could kind of tie into what we're seeing with our lab trials. So this is a more recent study from 2020, and Zorn et al. 2020. So they looked at Bay de Noc here in Green Bay, Lake Michigan, and found pretty low densities of zooplankton, fewer than 10 zooplankton per liter in this area. And I can tell you from the agencies that are collecting zooplankton from our field sites have found even lower densities of zooplankton here over at Manassik, and a few other sites around northern Lake Michigan are finding densities as low as 0.5 prey items per liter. So very reduced prey densities in this region. which leads to slower growth. And I want to tie in importance of cladocerans to lake whitefish diet. There's been a lot of findings that have suggested that cladocerans are quite important to diets in larval lake whitefish as they get to about above 17 millimeters in length. So they make up about from like a third to like half of the diet in larval lake whitefish, like in beta kinte as well. 32% of larval lake whitefish diet was consisted of cladocerans, and that includes Daphnia and Bosmina in particular. However, cladocerans have also been declining in abundance over the decades as well. A lot of which is due to dressidae mussel invasions. So once that occurred, already the assemblage kind of shifted in terms of size structure from bigger Daphnia to kind of smaller Daphnia and also Bosmina. And now we're seeing declines in those cladocerans as well. Which takes me to my third objective here, which is to evaluate the influence of prey type as well as availability on growth patterns of larval lake whitefish and Cisco in northern Michigan Huron sites. We also have one from Lake Superior as well. So we're back to our nearshore area habitat with our different prey items. So for our zooplankton sample processing, we have samples acquired from multiple agencies at various sites and that we were able to count and calculate density. And after density was calculated, we were able to measure lengths for 10 of each taxon per site and year. And we did this so that we could calculate the weight using published length weight regressions. For our larval fish sample processing, we measured length. We extracted otoliths and counted daily growth rings to get age. And then we dissected the guts to get all the prey items identified and shipped off the fish bodies to the USGS lab for genetic identification. And then so we calculated biomass of zooplankton using those weight length regressions and compared those across sites. We conducted age length regressions for larval fish. So age on the x-axis here and then length on the y-axis. We're able to get the slope to determine growth rate in millimeters per day. And then to compare diet composition between lake whitefish and Cisco, I conducted a non-metric multidimensional scaling ordination in order to visualize those differences, and then followed that up with the similarity percentages analysis to see what taxa were accounting for these differences in diet. So these are the sites where we've gotten a mix of both lake whitefish and Cisco. So Charlevoix and Elk Rapids are both in northern Lake Michigan, and Saddlebags here is located in northern Lake Huron. So these are our results for prey availability in these three sites. Elk Rapids and Charlevoix exhibited the most difference. Both are characterized by adult copepods, particularly calanoids and cyclopoids, mostly calanoids, especially in Charlevoix. And then in Saddlebags, there were quite a few adult copepods, but also copepod nauplii and chironomid larvae as well. So for growth by site, between lake whitefish and Cisco, again, we have Cisco in the orange and lake whitefish in the blue here. At each of the three sites, we see Cisco exhibiting a faster growth rate than lake whitefish. So here, Cisco grew at about 0.1 millimeter per day versus lake whitefish of about 0.06. Up here we have in Elk Rapids, we have a growth rate of Cisco at about 0.12 millimeters per day, as opposed to 0.1 millimeter per day for lake whitefish. And then again, and at Saddlebags, we have about 0.13 millimeters per day for Cisco and 0.08 for lake whitefish. And I also have the regressions that I have that kind of show patterns of when Cisco begin to start outgrowing lake whitefish. So in Charlevoix, we see that those lines intersect at about 16 millimeters in length. And same with Elk Rapids. We're seeing similar pattern here where Cisco begin outgrowing Lake Whitefish again at 16 millimeters. And then in Saddlebags, we see at about 18 millimeters is when Cisco begin to outgrow Lake Whitefish. So similar patterns throughout these 3 sites here in terms of growth. And then for diet composition by site, I'll explain this plot a little bit. These polygons represent each site. In the green, we have Elk Rapids. The red, we have Charlevoix. So quite a bit of overlap here in terms of diet composition. And then Saddlebags is in the blue here. And the squares represent Cisco, whereas triangles are Lake Whitefish. And I have the taxa names overlaid on the plot here. So the closer an individual fish represented either by a square or a triangle is to the name of one of the taxa we have in this plot, the higher abundance that was found in that individual fish. And so we're seeing this is from the similarity percentages analysis that generated some p-values and determined that there were significant differences between Lake Whitefish and Cisco diet at Elk Rapids. So Cisco had higher quantities of calanoids than Lake Whitefish at this site, as well as cyclopoids than Lake Whitefish. And then at Saddlebags, we see Cisco consuming higher quantities of copepod nauplii, as well as chironomid larvae. And that was the site that had the highest amount of copepod nauplii, as well as chironomid larvae. Now I'll talk about some sites where we only got Lake Whitefish. So that was in 2 northern Lake Michigan sites, Manassik and Brevoort River, and then one in Lake Superior, Big Abe's Landing. And I did this same process here. So this is prey availability throughout these 3 sites in 2019 and 2021. So Big Abe's Landing had the highest prey density in 2019, and that was primarily characterized by chironomid larvae. And this is just percentage composition at each site. And then Big Abe's Landing in 2021 had a reduced prey density, but also had a higher percentage of chironomid larvae than the other sites, as well as Bosmina. And then in Manassik, the rest of the sites, the densities are pretty low. And so Manassik and Brevoort River in 2019 and 2021, pretty low densities characterized by mostly cyclopoid copepods. As for growth of larval lake whitefish at these 3 sites in 2019 and 2021, we have the highest growth exhibited by lake whitefish at Big Abe's Landing in 2019. So that was the site and year that had the highest prey density. And then that growth rate is significantly reduced in the following in 2021, where we also talked about the prey being reduced at that site and year as well. And then Brevoort River and Manassik followed similar patterns here. So Brevoort River had pretty low growth at both 2019 and 2021, and it was reduced in 2021. And same with Manassik. And then over on the right here, we see how prey density has and larval fish growth has a very significant relationship. Prey density very much affects larval lake whitefish growth. So overall prey density was pretty low across sites for both 2019 and 2021. And mostly at Lake Michigan sites, we're mostly seeing copepods. And then in the Lake Superior site, we saw quite a bit of chironomid larvae densities in in Big Abe's Landing. And overall, very low Cladocera density. So density, we can see very much influences larval lake whitefish growth. Prey composition seemed less important, although that could be attributed to just how low the density was in all sites. If we had higher densities and they consisted of energetically rich prey items like chironomid larvae in particular, possibly Cladocerans like mostly Daphnia, maybe we would see maybe we would see a better correlation between prey composition and growth sites. So now I kind of want to draw some parallels between my field results and lab results. So if you remember me talking about the foraging trials that I did in the lab, small Cisco consumed higher quantities of Artemia nauplii than lake whitefish. And in the field, we were seeing higher quantities of copepod nauplii in Cisco guts compared to lake whitefish. And that was at saddlebags where we saw the highest quantity of copepod nauplii. Lake whitefish outperformed Cisco with Bosmina in the lab studies. However, there wasn't a whole lot we saw very low densities of Bosmina and cladocerans overall in the field sites. So while we can award Lake Whitefish the trophy with the Bosmina at all 3 size categories, not so fast. We're not seeing a whole lot in the field for it to benefit them. As for adult copepods, in the field, we saw small Cisco, so around like 16, 17 millimeter Cisco, consuming higher quantities of adult copepods in lake whitefish. So at Charlevoix and Elk Rapids. However, in the lab studies at that small size, remember there was no difference in capture and handling times between lake whitefish and Cisco at that small size. And then medium lake whitefish actually did have higher success with cyclopoid copepods than Cisco. And then as they got to that large size category, large Cisco had higher capture and lower handling times than lake whitefish. So there is a bit of discrepancy there between the lab and the field when it comes to copepods and the sizes at which they're consuming them at higher rates. So kind of to summarize everything I've talked about so far, because it all ties in together pretty well. So we're seeing a higher metabolism in Cisco at that smaller size. We're also in the field seeing higher quantities of prey items that we're finding in higher quantities in the field, so prey availability in the field, in Cisco diets as opposed to lake whitefish diets. That's it. We solved it, right? But is there more to the story than potentially Cisco having some sort of advantage over lake whitefish when it comes to prey and diet out in the field? And that's where predation comes into play. So I wanted to compare predation avoidance between lake whitefish and Cisco to these introduced nearshore predators, rainbow smelt and round goby. So round goby was introduced to the St. Clair River in 1990 and went up into Lake Huron and quickly invaded the Great Lakes. They alter benthic community structure. They'll eat a lot of benthic macroinvertebrates, benthic zooplankton. They can compete with native fishes for food. They also affect native fish populations in terms of eating their eggs, in particular lake trout and basses as well. Although there's not a whole lot out there about how they could potentially influence carrageenans. And there is there's a temporal and a spatial overlap with between the round gobies and larval lake whitefish and Cisco, since I mentioned that larval carrageenans will be found in those nearshore areas in the spring until early summer. And round gobies will typically start to come into the nearshore areas around mid-May and stay there until September or October. So there's about a 6 to 8 week overlap in habitat between the two. So that could have an impact on their populations. So for my predation experiments, I have this set up here with a camera stand, and this is a pair design. So in this tank, I would have round gobies feeding on lake whitefish, and then the one on the right, I would then have Cisco. So and each trial was filmed for 30 minutes. And as I'm going through the footage, I focus on 3 behaviors: pursuits, attacks, and captures, which I will show you guys actually in the next slide. I use 3 round goby sizes, small, medium, and large, and then 3 larval carrageenan sizes, also small, medium, and large. So I'm going to show you what I classified as each behavior. So this is a pursuit. So you see it focusing on this one fish here, this one, and it didn't get anywhere close to striking range. For attack, so you see how it definitely strikes, but then it eventually lets the fish go and he escapes and oh, okay. So I won't play my capture one. Okay, this is the capture. Boom, and so he successfully ingests the fish. So these are the three behaviors that I was looking for as I'm going through the footage. And so then I was able to calculate overall predation rates. So captures per goby for each trial, so an average, and then attacks per pursuit for each trial. So how many for each pursuit, how many attacks and captures are we getting? And then frequency of attacks resulting in a successful capture. And then for all these behaviors, I conducted a repeated measures analysis of variance. So this is my result. These are my results for the captures per goby. So a star indicates a significant difference. So from the left to right, we have larval fish size, small, medium, and large. And then from up to down, we have goby size, small, medium, and large. And you can see the large round gobies exhibited a significantly higher captures than on large Lake Whitefish than large Cisco. And for every other size combination, we did not see a significant difference. For captures and attacks per pursuit, once again, our small gobies didn't do anything interesting here in terms of differences between the two larval creating species. However, our medium gobies exhibited a significantly higher attacks per pursuit on small and medium Lake Whitefish than small and medium Cisco. And then our large gobies exhibited significantly higher attacks per pursuit on large Lake Whitefish than Cisco. For captures per attack or percentage of attacks resulting in capture, most groups had at least 50% successful attacks. However, at the medium goby size, we're seeing a significantly higher capture per attack on medium Lake Whitefish than medium Cisco, and the rest of them were not significant between the species. So to summarize this, the goby trials, we saw significantly higher captures with the large Lake Whitefish and large round goby combination, but other size categories, mostly the medium gobies, in terms of behavior, could potentially also favor Cisco at the small and medium larval sizes, since we're seeing higher efficiencies on larval Lake Whitefish than Cisco. And this could translate into the real world, right? So these 30-minute trials aren't exactly realistic. In the wild, they'll have all the time in the world to chase after these guys, and we could potentially be seeing a higher impact of round gobies on Lake Whitefish than Cisco field. So now I want to move on to rainbow smelt. So rainbow smelt were introduced to Crystal Lake in 1912 intentionally as forage fish, and then they inevitably escaped into Lake Michigan and invaded the Great Lakes. They spawn in the spring, so they'll run upstream, typically the second or third week of April. And the end of spawning typically coincides with hatching of larval crigatines. So they don't eat during their spawn. And so by the time they're done, they're pretty hungry and they have a buffet to chow down on, which are these hatching larval fish. So this obviously has the potential to impact recruitment of both larval crigatine species. And predation is directly proportional to larval crigatine densities. So you see a big patch of larval crigatines and smelter right there. They will have pretty high success. So I had the same process with the smelt as with the round gobies. I only used one rainbow smelt size range and then two larval crigatine sizes. And I classified each behavior the same for the rainbow smelt. So I'll show you each behavior. This is a pursuit. C is going after that fish, didn't get near striking range. And then for the attack here, see how it's about, it strikes this fish, it gets away. And then we have our capture. There we go. So these are, once again, the behaviors I'm looking at throughout the footage of each 30-minute trial. So these are my results for captures per smelt. We don't see a significant difference in captures between Lake Whitefish and Cisco, and size also didn't play a role or was not different in terms of captures. And the same for behaviors, so captures and attacks per pursuit, not seeing a difference between Lake Whitefish and Cisco at either size. And same with captures per attack. Once again, they have pretty high capture rate per attack for both species and at both sizes, but no differences between the two. So not seeing any differences for rainbow smelt, and size of larvae aren't significant. There have been other lab studies with rainbow smelt preying on larval lake whitefish, and this efficiency started to decline after the larvae reached about 35 millimeters in length, so maybe a little bit bigger than what I was using. And just in general, rainbow smelt just might not have the impact they once had on larval lake whitefish due to their abundance that has decreased. And so since predation is directly proportional to abundance of larval fish in the wild, we just might not be seeing an impact either. So I kind of want to show some behaviors that I saw while I was going through the footage here. I haven't quantified anything just yet, but I still was this pattern of behavior was so consistent that I thought it was worth showing here. So these are Cisco. And I tried to trying to show how tightly they stay together. So I'll play this again. See how they stay very close together and they're able to all move around together. Whereas with the lake whitefish, we're seeing them a lot more spread out. And this happened pretty consistently throughout these trials. So now I want to do something where I have you guys guess which species is which here. So I'm gonna go to my smelt trials and I'm gonna play, I don't have them labeled, so I'm going to show these video clips. And I actually want you guys to put in the chat which you think is which. So as you're looking at both of these clips here, see if you can guess which larval fish is in which tank. So if you want to take a second to put that in the chat. Yeah, a lot of you are getting it. So we're exactly Lake Whitefish are on the right, or sorry, on the left over here, and then the Cisco are on the right. So you see how those behaviors differ when avoiding prey. I thought it was really interesting to see that throughout these trials. And for round gobies, since they are a benthic predator, this could potentially make it easier for them to snag more lake whitefish, since it kind of make it easy for them to capture them. And for lake smelt, it might, or lake smelt, for rainbow smelt, it might not matter as much because they're kind of just up and down in the water column. And that might be a reason why there wasn't a significant difference in terms of capture or behavior between the two larval corrugating species. It's gonna let me change the slide. Okay, so that was a lot of information that I just threw at you. So I'm kind of going to try to synthesize all of it and talk about what this could potentially mean. So small Cisco, as we remember from way in the beginning, had higher routine metabolism. They were also found in higher abundance, and with at 2 out of those 3 sites that we caught, that we had a mix of both Lake Whitefish and Cisco, and then also with faster growth rates than Lake Whitefish at all the sites where we had a mix of both. All of these sites had prey compositions mostly consisting of adult copepods, and as we saw as Cisco grow in those lab studies, they became better able to capture adult copepods, although there was that size discrepancy between the field observations and the lab studies. So remember, the Cisco didn't have a higher capture rate of cyclopoid copepods until they got to that large size. So that could be, for a number of reasons, my current hypothesis is that there could be a gape size difference between hatchery reared larval corrugatines and those found in the field. So we're doing measurements on those actually right now. So we might have an answer for that soon. And also cladocerans once played a really important role in terms of diet for larval lake whitefish, and they had higher success with the bosmina at all size categories in those lab studies. However, we're just not seeing them in our field sites. So this could leave them at a disadvantage over Cisco. Seem to be growing faster in the field, and we're seeing higher quantities of available prey, which mostly consists of these copepods. And then for our predation studies, round goby predation behavior affected mostly medium and large lake whitefish significantly more than Cisco. So round goby could potentially have higher impacts on larval lake whitefish populations than those of larval Cisco as they get above 20 millimeters and above. And I think I forgot before to point out the fact that as lake whitefish grow, they become benthic. They'll develop a subterminal mouth and primarily be found at the bottom. Round gobies are also benthic. That could also play a role in affecting those populations through predation of round goby. And rainbow smelt doesn't seem like they play a role from the predation trials, since captures and behaviors didn't really differ significantly between lake whitefish and Cisco. And the fact that predation is largely associated with higher densities of larval lake whitefish, which we're not finding in some of these sites. So, but wait, there will be more. So next steps include, we're currently analyzing some data from a long-term growth and survival experiment that we conducted this past spring. So for these studies, we have single lake whitefish and single Cisco, as well as mixed species. So we're seeing if growth and survival differ between lake whitefish and Cisco at 2 different prey densities. So we're using a low prey density and a high prey density, and seeing if growth and survival differ and what could potentially contribute to that. So we'll be looking at diet as well. And then I'm actually here on Beaver Island all summer. So I will be collecting round gobies in the field and doing some dissections to see if I can get some data to complement what I'm seeing in the lab. So seeing if I'm finding larval corruganines in the diets of round goby in the field. And with that, I would like to acknowledge everyone who has helped on this project. So Sault Ste. Marie Tribe of Chippewa Indians and the Michigan Department of Natural Resources for field sampling for this talk. Lake Superior State University for sharing larval fish samples, the USGS Genetics Laboratory, Chris Day at the Little Traverse Bay Band of Odawa Indians for supplying larval fish, as well as Roger Gordon at the Jordan River National Fish Hatchery, Peter Dykstra for lending the respiration equipment, and Michael O'Neill for supplying larval fish equipment, and Ray Clark brilliant engineer who made the respiration chambers. And all the lab technicians who assisted with foraging experiments as well as field sample processing. Great Lakes Fisheries Trust for funding, and of course, Central Michigan University Earth and Ecosystem Science Program. And I'll take questions. I have my contact information here. I am graduating in a year from now, so shameless plug, always looking for postdoc opportunities afterwards. So feel free to contact me if you know anything through email, or my Twitter handle's there too. So I'll be taking any questions. Any.
Kelly, what a fantastic webinar. Unbelievably comprehensive, tackled a diversity of questions. So people, I'm sure if we were in a room, everybody would be clapping, so you can hear it. Maybe you can see the visual clap, the virtual clap there. But, and I love that you even did sort of the interactive. That's the first time we've done what a great idea. And so kudos for a great seminar. And I'm sure we're going to have a lot of questions. Feel free to raise your hand or pop your camera on, even better, so that if you have a question, you can also put your questions in the chat. I do have a hard stop at 2:00 today. But we could potentially go up to that time. And I know Kelly has the time also, for the next half hour or so. If we wanted to get into a discussion or just ask more questions about our great work. While people sort of think about their questions, Kelly, I wrote several down. I don't even know where to start. I'm amazed by the diversity of things you were doing in the lab, from the metabolism to predation to the foraging. One of the things that really stuck out to me is that plot you showed across sort of field sites where you had prey density, really explaining growth with a really high R-squared. And you did that really without thinking about temperature too, right? Because temperature could be really different in those sites, and we know temperature is a sort of master variable. But I don't know, I thought that was really cool to show that. And then also tying in that big Abe's Landing, that one Lake Superior site, how it jumped up so much in 2019. And that also corresponded, I think that was that one outlier point there.
Yeah.
But then but the next year in Big Abe's Landing, growth was down much lower. So I don't know, I guess, are you surprised that by the strength of that relationship you showed across those 6 or so sites, and how important?
I'm not surprised.
To growth? Yeah, go ahead.
Yeah, I'm not surprised by that relationship. We even eliminated that last Big Abe's Landing in 2019 to make sure that the relationship was still going to be strong, and the R-square was still about 0.7. So, which I pretty much expected density to have a pretty big effect on growth. So I wasn't surprised by that. I was surprised by the drop in density from 2019 to 2021 at that same site, but since it was so pretty dominated by the chironomid larvae, I almost wonder if that sampling year was just different in terms of like how it was sampled, or how many chironomids ended up in that sample, because it wasn't exactly, we don't exactly like, we use zooplankton nets, so I was a little worried about how that could play into it, but I wanted to include it just because I do believe that chironomids do play a pretty large role in diet and in growth as larval fish. So
okay, Erin Dunlop has her hand up.
Great job. Such cool stuff that you're doing. Yeah, it was great to see your stuff at iAgler, and then this was, even kind of like, I loved how you put everything kind of together and have a really cool story to tell. I have several questions, but I guess one of them is kind of related to what Bo said, which is about temperature, because we do know that like temperature and spring warming rates affect larval growth quite a bit. So I was just wondering if you had any thoughts of, incorporating like growing degree days or some kind of measure of temperature when you're looking at growth between Cisco and Whitefish and the different study sites and years?
Yeah, it's a good question. So I definitely do want to incorporate temperature. I need to get that those data from the agencies that have been collecting these samples, and I really do want to. It's been something that's been on my mind for this entire time. I just haven't gotten around to getting it yet, but I do definitely want to incorporate temperature. And I try to make sure that the sites that I'm including were around the same time, so that there wouldn't be that I guess confounding variable there. But temperatures like of course could vary from year to year as well. So I would love to get that temperature data and incorporate that into that as well, so I can kind of sort of, that would definitely make the picture a bit more inclusive when talking about how that could affect growth, because yeah, it's very important.
Yeah, cool. And just maybe one other question. I don't want to hog it, but maybe I'll just ask one more. So your experiments, your predation experiments, really cool again. Like it was really neat seeing the Cisco kind of school up. Like, I mean, they're schooling species, right? So it's kind of cool to see that already at the larval stage, they're sort of showing some signs of that. What I was curious about was how old those larval fish were, because I mean, it was just really cool to see what capable swimmers they were and how responsive they kind of were to the predators being there. And, you know, I think a lot of people think of these larval fish as just sort of like kind of floating around and getting moved around by currents and all of that kind of stuff, but they seem to be really capable active swimmers. And so I'm just kind of curious how old they are, because, you know, I kind of wonder, like, did they start out like just sort of being a little more passive and then they sort of develop their swimming capabilities as they age? Just something I'm curious.
Good question, because so I did have 3 different sizes, and the reason I showed the larger size is just because it's easier to see, especially for a webinar like this. But I did notice that in the first with the smaller larval fish, so probably about like like 3 or 4 weeks old, because they hatch at the hatchery in January, and then I would start trials about like a couple weeks later, so 2 to 3 weeks, and then it takes a while to get through them all, so they're at least a couple weeks old, and then the larger ones are probably around like 3 months old. And so the small size category of larval fish, I didn't see that pattern as much. And it was when I got to the medium size category, so like 20 millimeters and above, is when I started seeing that difference as far as like schooling behavior in Cisco, but not as much in the lake whitefish. But starting out, they were both kind of like swimming all around and not really close together like we saw in the video clips.
Yeah. Okay. Yeah. Thanks so much.
Yeah, of course. Yeah. You see Taylor's hand up? Okay.
Yeah.
Thank you. Okay.
Yeah. Okay. So yeah, great presentation. I'm really curious about the aging as well, since I know a lot of people aren't aging Cisco and Lake Whitefish in the same place at the same time. And there's a lot of uncertainty about when exactly they hatch. Does Lake Whitefish hatch first? Do they hatch at the same time? What's the actual difference? And so I'm wondering if you were able, with your aging, to get a handle on that and try and understand that, because especially if Lake Whitefish are hatching out, 2 weeks earlier, let's say, they can be really big already by the time that Cisco hatch. And so that would have pretty significant implications on some of your results, I think. So I'm really curious if you looked into that.
Yeah, so it looked like from the regressions, and I can go back to that if I can. Right now my screen is black. Oh, okay. I can go back to the regressions, but yeah, it did seem like the Cisco were starting out smaller than Lake Whitefish.
Yeah, from what I've seen, they definitely hatch out smaller. But yeah, I was curious, like day of year when you started, like when you back calculated hatch date, for example, if you saw any differences there, and if so, by what magnitude?
So I didn't
Kelly,
back calculate.
This is Jory. Can I just kind of address it a little bit? I think that's one of this data set is so rich. And this is one of the things we need to use it for. We haven't asked her to use it for that yet, but there's just so many great questions that can be teased out of here, and it will be relatively easy to do because we have a capture date and then we have daily growth rings. So yeah, I think it's a great question. And it would be nice to swing back around and do that at some point. So.
It's something that Jory and I talked about, back calculating those hatch dates, but I haven't done that yet. But from these regressions, I mean, they're pretty much hatching, like you see such variation, we see such variation in these ages, and it just kind of goes to show like they're just hatching all the time, like just throughout the spring. And so I think that's where a lot of that age variation comes from. But I do want to do back calculations to hatch date, and because that would answer that question a little bit better. But
yeah, that's
yeah,
super interesting. I knew it wasn't something you directly reported on, but it just seems like one of the many things that you can do with this data set.
Yes.
I will say that for Lake Ontario, we see that Lake Whitefish continue to hatch at the same time that Cisco are, and you're right, it seems to be some sort of, there's a lot of different cohorts going on, and so I'll be curious to see if that was just a weird year or if that's more consistent. Thanks.
Yeah.
So there's a comment in the chat from Wendy Stott. The differences in capture efficiency between field and lab are interesting. And then she's talking about in some work head shape differences between wild and lab that she found in her lab, that they could start very small sizes, like 30 millimeters and smaller. And then she also did some experiments where she found that fish reared at different temperatures sometimes could be more like a wild fish. And so just bringing up, I think, some of the points about, to Aaron's point, the temperature inclusion, exploring some of that. And then also just what you mentioned too about looking at the gapes between the wild fish versus the hatchery fish, because those hatchery fish can sometimes have those malformations in the head. And then she also noted, and I don't know if you've looked at this, the eye shape. You see that's comment that she made. And so if eye size, for example, happens to be larger, so.
That's a good point too. Yeah, I haven't looked at eye shape, but that's a really interesting point. Definitely.
Did you want to add anything else to that, Wendy? Like via just the voice here? Okay.
Oh, Aaron. Oh, you're muted.
Sorry. Just a follow-up question on this slide here. So you have age and days, and I'm kind of curious if that's actually, if it's number of increments or actually age, just because sort of some of the work we've done with whitefish, we have found that like the check and sort of when they start forming those increments occurs about like after day 20. After they're 20 days old. And same with the Cisco, some of that earlier work you probably know of, where they also found that the increments are formed at about day 20. So kind of just wondering, are these ages or are they number of increments or rings?
Number of rings.
Okay.
Yeah. So
Yeah.
I have heard there's a lot of different, I guess, issues with counting daily growth rings and finding finding hatch date was obviously the most challenging for us. But once we standardized that, we're pretty much on our way. But then there's other things that I've read and that I've heard from other people on this call actually that makes me kind of want to go back and look at those again. But
Yeah, I mean, I
these are-
think
these are
yeah, I think so. We like actually have a paper just that just got accepted that's coming out where we did some aging validation for whitefish. So I'll be happy to send
Yeah.
it to you. But I think
Great,
it
yeah.
just means like when this is just a suggestion, but like with this kind of graph, even just if you just had increments there, that kind of takes care of it. Everyone knows what you're talking about because it's, or it would be like post increment age or something, right? Because it's not the true age of the fish, because like as Taylor was saying, like they hatch, they have their yolk sac, and it's a certain number of days before they use up that yolk sac and begin feeding, and that's when the increments start forming. So like I say, they could already be 20 days old before they start forming those increments. Anyway, just something to think about.
Thank you. I think I see Zach.
That's awesome. It's nice to see you again, Kelly.
Hi.
I guess I have a comment on what we were just talking about and a question. So I've been obviously doing a lot of the same stuff, and I've aged 3,500 larval corygonids that I caught out in Lake Erie. And thinking about that increment counting, well, Aaron, are the ones where they're not laying down rings until 20 days after, are those from hatchery caught fish?
They are from fish that are from wild parents that are raised in the lab. But yeah, they're in a lab environment. And then in this particular study, we also compared it with some wild caught fish as well.
In the lab ones, the incubation period is relatively short compared to the wild fish.
No, it's actually pretty similar. So because it's a naturally fed, the water is coming from like a lake, this lake fed. So they're hatching at about the same time-ish as they would in the wild.
I've been very curious about all that too, trying to figure it out.
Yeah.
One of the things I did after I did back calculate my ages to see like when I was getting fish with 30 or 40 increments, look back to when I was sampling during that time period to see if I was getting larval fish at that time. Because if you're getting fish that are like have 60 increments, you look back 60 days ago when you were sampling and you weren't catching any fish, then that might be an indication that something's a little wonky. So that's one way that I've kind of helped check myself. But I'm curious about your third chapter. I think you ended with a comparison statement between your lab stuff and your field stuff.
Mhm.
What was that statement? I'm trying to remember all of the graphs, and I could be wrong, but were your densities for the lab experiment measured by number of nauplii and number?
Yeah.
And then for the field, weren't they converted to biomass? So where in the lab you could be finding a lot of nauplii in the stomach. In the field, that's like a relatively small percentage of their actual diet.
Yeah, so I did, for the foraging trials, it was we measured the densities in number per liter, and then for the field, I was going to stick to numbers per liter, and then kind of I decided I should probably convert to biomass, micrograms per liter. So before I converted that, I was finding like upwards of like 40 nauplii in a single Cisco gut. But when you look at it in micrograms per liter, you're right, it doesn't contribute very much in terms of biomass. So I was kind of thinking about how I want to standardize that. Maybe I will convert my lab densities to biomass as well. To kind of.
We're seeing the same thing in Lake Erie, the same trend. And one other thing that instead I didn't measure, it looks like all of these are pretty near shore sites that you collected.
Yeah.
I collected near shore kind of, and then, incrementally offshore, off to like maybe 10 or 15 miles offshore. So I measured in a row, and I measured the zooplankton as we went out and what each fish was eating as we went out. And in those offshore areas, we were, at least in Lake Erie, which is totally different, we were getting larger fish. So the fish near shore were actually much smaller than the offshore fish. So they would start to develop fins at like 17 millimeters and then just leave those near shore areas and go offshore.
Yeah, I
And-
remember you saying that about like how much smaller they are when they go offshore compared to Lake Michigan. Yeah.
And they were eating the same amount in like those offshore areas with very few zooplankton. They had equal amounts of food in their stomach as the nearshore plankton with tons of food available. Relatively speaking for Lake Erie, which is much more productive and all of the things that go along with it. But just an interesting comparison that I noticed.
Sure. Yeah.
So Zach, just to be clear, you were getting I mean, I'm guessing these are whitefish because you're in Lake Erie. You were getting whitefish larvae 15 miles offshore in the pelagic zone, or
Yeah.
wow.
Yeah.
And
Every
maybe we'll
year.
have to book you for a webinar this fall.
This fall would be good timing. I plan on graduating about then. So
Yeah, that's
Um,
interesting to get that, because one thing that we've been trying to, I mean, this like Kelly's work and a lot of the work is sort of beach oriented or small vessels closer to shore, but finding them offshore, we've looked and haven't really been able to do that. So that's gonna be interesting to learn more about.
yeah, and they're at the surface offshore. So regardless of where I sample, they're always at the surface. I've sampled at the bottom throughout the lake and they're just always at the surface, even when most of the offshore food is at the bottom during the day. They hang out at the top.
Mhm.
And following up on Aaron's comment, I find very few fish, even with full yolk sacs, that with empty stomachs. So at least in Erie, it's relatively rare to find a fish that's not feeding, even 13, freshly hatched, 13 millimeters, that sort of thing. Definitely less than 10%.
Sac, same here. The yolk sac does not preclude feeding.
Cool.
Okay, thanks, Zach. And
Yeah.
some more information on the chat. Wendy posted some images. Her mic isn't working. Taylor provided another link to an age validation study. And then made the comments about the different basins of especially Western Basin Lake Erie having a lot more potential prey density than say some of the offshore upper lakes. And Aaron, just recognizing the diversity of all these prey sets, data sets, just talking about the value and potentially pulling them all together at some point, learning more about the early life history feeding. So one comment, or I love that you did this sort of these, you called the predation avoidance experiments, Kelly. And I'm thinking now, like from a conservation hatchery perspective, where, you were dealing with hatchery fish. Most of the hatchery fish have no exposure to predators before we put them into the lake, and they're going to be exposed to predators right away. Obviously, mostly at larger sizes because we stock them at larger sizes than maybe what you were doing, but they figured it out. Like they seem to be, at least by the 20 millimeters, the schooling behavior you talked about. And I wonder, like thinking about how this could potentially transfer to a hatchery setting to try to expose some of these animals to more real life conditions. Was there one that was worse at actually eating the fish, but could still like scare them and think that a predator is present, and to sort of develop that? Like it was hard to tell between like gobies and smelt in terms of which was better at sort of capturing prey. Was goby a more effective predator than rainbow smelt, or have you looked at
That's
the
a
data?
really good question. I felt like with the smelt, it was a little bit more variable than the gobies. It was with the differences in behavior between the predators, I noticed was that the gobies seemed to kind of like just sit there and wait, and as something would come along, go and grab it. And that's why I feel like as the Lake Whitefish grew bigger, they're kind of more below towards the bottom compared to Cisco. And so it kind of made them easy targets. Whereas for smelt, they kind of are all over the place. They're swimming up and down in the water column and able to navigate pretty well. And so yeah, the differences in terms of the predator behavior themselves, I feel like contributed to those differences. But I did see also that when gobies did kind of like move towards like a school of Cisco, they would all immediately swim off together, and the goby would kind of give up. And I saw that happen a lot. Whereas with the smelt, it was I feel like they had an easier time. I felt, yeah, in general, kind of going after both, kind of with their ability to move up and down the water column the way they could.
Okay.
So
Again, really cool experiment. Glad that you did it and helps spark other ideas. So
yeah.
Corey's got his hand up. Corey Brandt.
Yeah, Kelly, so awesome. The research, the diversity of stuff you're studying. I was locked down the whole time. Bo kind of sparked me on this question. I was kind of sitting excitingly with it. Regarding the chamber experiments and the behavioral trials, I find that work very exciting. And the stuff that you're noticing with these very young larvae is really notable. And so my first question is, do you plan to continue some of that and maybe quantify some things? I hope so. And then secondly, in laboratory experiments, in my experience, in those experiments, the behaviors in some trials really significantly changed when some enrichment was added, perhaps some substrate equal amounts across chambers or different habitat types. And so it could be, if you have time, which is maybe a pipe dream, I don't know, it'd be interesting to test different environments for these species. Perhaps you're finding that instinctively whitefish disperse and cover, while Cisco very early on have this instinctive shoaling behavior. And this could inform where these are stocked in a big way. Perhaps Lake Whitefish better off in rocky areas versus Cisco in more open areas where their schooling behavior can benefit them. So it's really exciting. And yeah, I'd love to hear your thoughts on it.
It's a good question, because I actually have conducted trials with substrate for the predation. I haven't gone through them yet. I do want to go through them to see if those behaviors are different. It just, at the time, it wasn't a priority. I was just trying to get through these experiments that we had already originally proposed doing, but it's something that I want to look at, those trials with the substrate. Although what I did for that is I took some cobble and I kind of put a little wall of cobble around in the tank. So that's what I did in terms of substrate. But if there's like another way to involve substrate in that, I'm always open to suggestions. But I kind of want to go through those trials first to see if behaviorally we're seeing what you're mentioning, differences in avoidance patterns there.
That's awesome. Seems like an exciting start. Yeah.
Yeah. I was like, let's see if it's different without substrate first, and then kind of go from there. So, and maybe if I have time, I might do more of those substrate trials, and see. But what was your first question? I think I missed something. I think I missed one. Or did I answer it?
I think I was just more excited. I don't remember.
Okay.
But I think you answered. I mean, my first question was, did you plan to continue that work if you have time? And it seems
if
like
I have
yes.
time, possibly. Yeah, I
Yeah.
do want to go through the existing trials that I have. But as far as doing more, maybe not now, at least for this, getting my degree, and then potentially later on, if there are opportunities to continue with that, I would love to, because it's been very exciting. Gotten myself down a lot of different rabbit holes. Wasn't even planning on doing metabolism until I found out some information and decided to do that. So it kind of as I advance in my career, I would love to continue on with these things. There were some things that I had to tell myself that I just can't do it, even though I want to, because there's just not enough time. So
Yeah, that's very reasonable.
yeah.
Sounds like you might get a metabolism question next. I see Kevin Keeler.
All
with his
right.
hand up.
No. Yeah, you're right. So curious about that part of it. What was the size of that chamber, the volume of it, and then what was the size of those fish? Not in terms of length or anything, but in terms of, weight, grams that you were able to fit in there. It was great to see that the controls, your background respiration rate was just, flat, so you don't have a lot of bacteria. But how did you decide on the number of fish to go into there? Because you definitely have too few fish where you can't get any readings, but you can also have way too many fish and they stress each other out, and make that O2 go down way too fast. So just curious about that sort of setup.
It's a great question. So the chambers were 22.5 milliliters in volume. And so I kind of went from there. We have an amazing engineer at CMU who made these chambers. And then there's a paper, Moyano and Peck, I believe 2016, that talks about different respirometry methods. And there is a kind of a ratio of chamber volume to size of larval fish that is appropriate to determine how many fish to use. So for the small fish that were about wet weight, like 0.00, like 5 grams on average, I think, if I'm remembering correctly. I have it all in my files.
Did you have a single fish in that chamber?
No, in the small fish chambers, I had 5.
Okay.
And from that paper, the 2016 paper, the ratio that they suggested was for the mass in, like, how much like mass in that chamber should be about 200 to 300 times the volume of the chamber. So I weighed out a bunch of fish, got the average weight, and then calculated how many I would need to get a detectable oxygen consumption. Because I did think about, like, oh, I don't want to stress the fish out by adding too many. So I kind of did the lower end of that ratio for that reason. So I'd be able to get a detectable oxygen consumption without adding more stress than necessary to fish in there. So, and then for each trial was an hour long, and I took the last 20 minutes to 30 minutes of those readings to make sure that I'm not just getting the beginning part where they're in the chamber stressed out, which I've seen in other studies as well. So.
Okay, great. And you definitely never use one fish.
I used one fish for the large size because I did the same thing where I weighed out a bunch of fish at that size, about like between like 0.08 and 0.1 gram wet weight, and was able to determine that one fish would again be on the lower end of that ratio, but still give me a detectable oxygen decline.
So as they're growing, you're using less and less fish. Okay.
Yeah. Mm-hmm.
Okay. And I mean, well, I can ask you another. I think I'll hold my question just in case there's one more, because we're getting really close to that 2 o'clock time where I know at least I have to go. But we're still at over 75% of the audience almost staying on here for all these questions because they're
Thanks,
interested,
guys.
Kelly. So it's a great job in doing the presentation, and sort of I think everybody's enjoying hearing more about it and your work. So
Yeah,
kudos
feel
to
free
you
to
and
reach
your
out.
co-authors. Yeah. Any final questions or comments? All right, well, send Kelly a follow-up question if you have it. You have her information there. Twitter and email, And thanks again for everybody for joining us, and we'll see you back here again in September.